Thank you. So thanks a lot for the invitation. It's really an honor to be here, and I'm looking forward to the discussion. My name is Georg. I'm from Medical University, but I'm a mathematician, and we are a group of about 30 people who are part of the radiology department. We are interested in how we can use imaging data to understand a bit more about disease, about the progression of the disease, and how we can inform treatment decisions. And today I'd like to talk a bit about what we learned about the brain, so what we learned about how the brain reorganizes during disease, like for example during cancer when it tries to compensate for the loss of functionality in certain brain regions, and how we can use some observations about brain development to understand it better, and ultimately how that led us to trying to understand how brain evolution plays out and what we can learn. There are two things that I'm really curious about that are connected with that. One is map building, so the idea of capturing, observing lots of relationships and putting them into a representation that you can learn from, into a representation that you can look at and that tells you something about the overall structure of what is in front of you. And Xavier and me were just discussing a bit about the conferences that we go to, and one of the most impressive moments in the conference was in the IPMI conference, which is a bit of a unusual conference because there are no speaking time limits and there are also no discussion time limits. So the most fierce and most emotional discussion I ever witnessed was at that conference, and it was going for 1 hour. And the discussion was about the matter of correspondence, about atlas building, about how we can tell what is the same point in one brain and in another brain. Because a lot of what we know about the brain and a lot about what we know about neural architecture stems from our assumption that we are able to say, well, this is the same point in the brain of one person and in the other person. And so part of this talk will be about how we can view that as a problem of map building and how we can actually say that there is not a single correspondence between different brains, but there are different correspondences depending on whether we are looking at anatomy or function or other characteristics of the brain. The second area that I've become a bit more curious about lately is some aspects of church architecture. And one of the aspects is these little triangles. So does anybody know how these triangles are called that you see on the four corners of this inner space? So they are called spandrels. The special thing about spandrels is that when you are basically ordering an architect to build a church, you're not ordering the spandrels, but you're ordering basically, well, that's my kind of base layout of the church, and I'd like to have kind of a spherical cupola on top of it. If you do that, then you need something to fill the holes that are emerging when you're putting these two geometries together. Nobody wants these spandrels to begin with, but eventually you realize you need them in order to kind of make the church roof a hole. And what happened during history was that once they were there, people started to use them, and they became a very specific part of the artwork in churches. And so during the talk, when we are talking about brain, about evolution, about reorganization, we will touch these two ideas about correspondence, about map building, and about a couple of features of the brain that are a bit like these spandrels that happened by accident, so to say, or that might have happened by accident, but took on very interesting roles in our current brain architecture and cognitive ability. So we'll touch these 2 points a couple of times, so keep them in mind. These spandrels that are there by accident, and the idea that correspondences are not something that is just a unique relationship between two brains or two observations, but there might be multiple of these relationships. So I'm coming from medical university, so a lot of our motivation is in the clinic. And one of the problems that we are trying to tackle is that there is a big population of patients who have a tumor in the brain. So what you see here is kind of an MR image, so magnetic resonance imaging data of the brain, and you see that there is a little bit kind of irregular tissue here, and these are brain tumors, and they are labeled on the lower part. And it turns out that for patients who have tumors that grow, for example, into the language center, for some of these patients, the language keeps being intact. So even though the area where the language was residing is destroyed by the tumor, it turns out that they still are able to speak, they still are able to understand. So the language center has to move somewhere else. That is the first point where this assumption that we can kind of map between brains by saying, well, that's the same point on the brain, so it must be approximately the same role. This relationship that we base a lot about what we understand about the brain, where are the language center, where the motor center, where the vision centers, about that kind of breaks down in this situation. Because when we are looking at these patients, we realize that the language networks that are connecting speech and language understanding and language production have moved to different places. Right, so using anatomy as a basis of our map of the brain and as a basis of saying, well, that's the same location in this and the other brain, does not work anymore. But as you know, probably much better than I do, when we're talking about maps, when we are talking about manifolds, they are essentially just representations of relationships. And so when we are talking about the anatomical map, that they are kind of summaries of relationships in geographic or in spatial space. But we can also look at other relationships. So we can look at networks in the brain. And one way to look at networks in the brain is to observe the brain over longer time periods and look at the signals in the brain and then look at the correlations between these signals. And these correlations give us some indication whether there is a strong or a weak relationship between 2 points in the brain. And so we can imagine that as a big graph that connects different points in the brain. We can write that graph down as kind of affinity matrix that tells us about the pairwise relationships between different points. So that basically every edge in this graph is represented by one entry in this affinity matrix. Then we can use any of the many manifold learning methods to basically embed all of these points into a new space where the Euclidean distance represents the distance, the travel time through this graph. So we used diffusion maps in order to embed all of these points in the brain into an embedding space, so that in this embedding space we can just use the Euclidean distance between 2 points, and the Euclidean distance represents the travel time from one point to the other to this graph. So the Euclidean distance in this embedding represents how closely related these 2 points are acting when we are observing the brain. That's a bit different from the spatial relationships, right? Because sometimes very far apart regions are talking to each other. We come back to that, how we can use it in order to track this reorganization in brain tumor patients. Yes. That's true, that's true. So these are very, I'm sorry, so these are very kind of abstract connections. We just say, well, these connections are just representatives of how strong the correlation of the signals is between these 2 points. We learned that there are, of course, also anatomical connections in the white matter tracks that are physically connecting points on the gray matter. And some of these direct connections actually correspond to these curved connections, but many of those do not correspond to any of the connections that we see in anatomy because they're basically kind of mitigated multi-jump connections. Thank you. So I need to clarify that. So these are really just functional relationships. There is some association with the anatomical relationships, but it's not the same, especially during disease. But they are just in this visualization. They are just tell you about the distance. So they're all the same size. They're just used as kind of a perspective. No. I just wanted to get a grip on how this. Okay, so that's kind of just the schematic. In reality, it looks somehow like this. So on the on the right-hand side, you see the cortex of an individual, and on the left-hand side, you see the embedding. You see basically this when you do a diffusion map embedding of these functional relationships, then all of these points get a representative in this functional space. That's just from one individual person, where the Euclidean distance tells us something about how strong the functional correlation is in the signal data that we get. These are just 2 dimensions, so it makes sense to look at more dimensions, but already in the first 2 dimensions, we see quite a lot. Now we're talking about correspondence, about maps. You have 2 brains, you can say, well, I can fit them on top of each other with geometric deformations and say, well, that's the back of the brain in 2 persons, that's the front end of the brain. And a lot of that works quite well in order to capture these relationships. So what do we do in the case of these cancer patients where that doesn't work anymore? It turns out that these maps are actually stable enough so that we can compare them between different patients. So what what I try to visualize here is we have two different persons. In the middle, you see a pair of two embeddings of these functional relationships of these two persons. And now it turns out that these are similar enough so that we can turn and deform them to find matching areas in the brain that are behaving similarly in terms of connectivity to other brain areas, no matter where they are. And if you're looking for an analog, it's like the brain is the map of Europe in geographical distances. This is the map of the brain, like the map of Europe when you're talking about, let's say, travel time by train between areas of Europe. The key is that these are stable enough in order to match them disentangled from the geometric space of anatomy. We need that for the brain. Yes. Why we assume that why it makes sense, or oh no, sorry, no. So I just visualized three dimensions or 2 dimensions. Like we usually, the number of dimensions that we use before it kind of dissolves into noise are maybe 20, 30 dimensions. Yeah, absolutely. Yeah, there is no correspondence between 3-dimensional space in anatomy and function. Right. Yes. No. So one way to talk about the correspondence would be to assume that the correspondence on the kind of roughly matched cortical surfaces counts and is correct. But I can also forget about this correspondence to just the embedding into the embedding space, however many dimensions I use, and then look at these point clouds, and then look at how these point clouds might be matched to each other. And kind of the coarse structure is similar enough so that we can actually do that. In this space. So okay, yeah, good question. So here for the anatomical space, we use something that's called T1, weighted magnetic resonance imaging, so MRI T1, which tells us something about the tissue properties. But for the functional maps, we are looking at essentially oxygenation signals in the brain. And the reason we do that is that there is a mechanism that when there is activity in a part of the cortex, this part of the cortex signals to the vessels, to the small vessels around it, to dilate a bit because it needs oxygen. And this dilation leads to the fact that in this volume unit, so we are nowhere close to microscopy, let's say 1 by 1 by 1 millimeter, but in this volume unit, you get an increase of oxygenation. We can see that in MRI. It's happening a bit later after the actual kind of activity, let's say 2 seconds later. But we can see these fluctuations of oxygenation. And when we see them at different parts of the brain, we can just look at the correlation. There is a lot of very good criticism about the limitations of just looking at the correlation. You might see a lot of things that have nothing to do with relationships in the brain, like with cardiac cycle, right? The cardiac cycle is a big signal in this correlation. But a little bit of these correlations seem to be correlated to neural relationships. Yes. Yes, resting state. Actually, in practice, so for research in an ideal world, we use resting state. For the tumor patients, now they start to do resting state, but before that, they usually did language mapping. So they put them into the scanner and they had some language paradigm where they asked them to speak or listen to something. Because the primary motivation to do that is to figure out where the language centers are so that they are not destroyed during the surgery. But it turns out that you can use that almost as well as resting state, because a lot of these correlation patterns, the big power of this correlation is actually similar to resting state. And the language paradigm, what we actually do is just a little bit of additional signal on top. Yes. So the voxels in this data are typically, let's say, 1 by 1 by 1 millimeter, 2 by 2 by 2 millimeters, so nowhere near neurons, right? So it's really a summary of a small region. Good. Yes. Yes. And it turns out that the first couple of axes are pretty well aligned to start with. Exactly. It's exactly that. So right. So let's have a quick look at what we can do with it, at kind of tracking these networks. So one insight is, for example, this is a patient where we have marked that's a glioblastoma, a brain tumor that's very local and a kind of tumor, and around the tumor there is edema, so kind of starting to the blood. This is in an anatomical space where we can look at the tissue and see that it's different from how the brain tissue should look like at this position. Now we can also look at this embedding map and see how that becomes deformed. And we can look at how the normal variability in a normal control population, kind of what's the variability there, and where do these different brain regions go out of this kind of scope of the normal variability. So we can think about set values. We say, well, here in these blue and red areas, there, this functional connectivity to other areas is starting to be out of the ordinary, stranger or very different than we see in the variability of a normal control population. And this gives us a tool to look at the sequence of what happens while these patients are treated. So what you see here is the same patient, a time series. That's the anomaly of these functional networks before surgery. Then after surgery, so we still see a lot of anomaly, mostly because of kind of swelling and very mechanical occurrences that happen after surgery. But eventually that goes away. Then there is radiotherapy happening, and we see how this anomal kind of connectivity network wanders a little bit back, relationship pattern in the brain. Right? And this gives us a tool to understand not only what's happening anatomically with the tumor, but also how the entire brain is affected by this. Yes. Yes. So we define it by every point has kind of distances to other points in the embedding. That gives us a profile. And we observe this profile in, let's say, 500 persons that do not have tumors. And we look at a new person and look whether that is in the distribution or how far outside of the distribution of these 500 normal points. Oh, the connection is like the spatial the distance in this embedding map, right? So we basically look, we encode, so to say, the position of one of these points as the distances to, let's say, a set of other points. This gives us a distance profile, and we look at this in a control population and then compare one to this. It's actually pretty well clustered. So we were surprised how well clustered it is, because we needed to collect data from different institutions to get a sufficient size. And we were actually afraid that that would make it impossible, but it was pretty consistent. At least at the resolution that we looked at. Right. Okay, and so now the next question is why are these areas starting to be not normal? Because when you look at where the tumor is, and when you look at where these not normal areas in the functional architectures are, it's not really that those are the areas that are close to the tumor. But it turns out that when you're looking at the distance in this functional space, these are areas that are close in the functional space, plus minus noise and plus minus a lot of factors that we don't understand, but largely the distance in this functional space, that's the good predictor about which brain areas are affected by a tumor compared to the distance in an anatomical space. So it helps us to understand a little bit the relationship and how this relationship pattern is affected by tumor. One observation that we made in this cohort, and it's definitely a preliminary observation, but we observed these patients over a long period of time. And sadly, many of these patients do have recurrence. So the first tumor gets removed surgically, but eventually, after some time, there might be a new tumor. And when we observe these patients over time and look at how these functional relationship patterns or kind of these profiles, however we represent them, but these relationships change, and we see that they go back to normal, and sometimes they'll go back to not normal. So things change again. That's this red dot on the top. And at this point, we did not see anything in the anatomical span. 2 months later, we do see the reoccurrence of the tumor. That's a small cohort of about 15 patients, but here we see consistently, if we see a reoccurrence of the tumor, we see this anomaly before. When you're talking with medical experts, then of course this anomaly might have different reasons. So one might be reorganizations, but one might also just be that before the tumor is occurring, be visible in the imaging data, you already see vascularization changes in the blood flow and in the blood supply in these areas that we don't see in the anatomical scan, but that we do see when we are looking at this oxygenation. Right. Yes. They are occurring near to where the new lesion emerges anatomically. Oh, true. No. So we did not look if the new lesion occurs at the near space in the functional space. But I totally agree that that would be really interesting. What we do see, though, is that it often actually occurs close by in the anatomical space, because sometimes it's just that there was a little bit of residual tumor left after the surgery that eventually comes back. So then the next question is, we see that in this patient population, but what happens in the normal population, right? We now have two ways of saying, well, that's the same point. That's the same point anatomically, and that's the same point functionally. Is there some difference between these two, and is this difference somehow does it tell us something about the brain? That's an older paper, Sophia Müller and Hesheng Yu, and they looked at the overall variability of this connectivity profile. This is the map, and the red areas are very variable. The blue areas have very little variability, just in terms of how they are connected to other areas, and we come back to that a little bit later. And the blue areas, for example, this is the motor cortex, right? So where you steer movement, where you're feeling things, the back part. The blue part is the vision cortex. So these are relatively old areas evolutionarily. The orange parts, these are parts that are very comparably modern. So they have gained a lot of volume during the later stages of evolution. These are also the ones that are much more variable. We'll come back to that. We are talking about the evolution. That is in anatomical space. So when we do the same thing that we did before for the patients, for a big population of, let's say, 500 individuals, that's worked by Daniel Margulis and his colleagues, then we can map this entire population in this functional space. We align it there. And of course, it loses a lot of detail, but we get an overall very dominant structure of this three kind of poles in this distribution. When we kind of visualize how these three poles map back on the cortex, and we see that we get these two poles in this functional space that are corresponding to visual cortex and motor cortex. Then we have these red areas that are another pole that are mapping more to something that's called default mode network, to the frontal cortex, again, to more modern areas. That's if you're looking at the group. I think the interesting thing is now if you're looking at the variability within the group, right? Everybody has a little bit of a different brain shape, but might also everybody have a little bit of a different assignment about which brain areas are responsible for what. So we did an experiment with data of FCP data set of 100 individuals, and the good thing about this data set is that we have multiple scans. And what this shows is when we scan a person 2 times, then we do the registration, so finding correspondence based on the anatomy. Then we take the functional data and perform correspondence based on the functional data. Do they agree or not? Do we get to a different point on the cortex when we follow the functional correspondence compared to the anatomic correspondence? And when we do that, let's say for 100 persons, but every time we only compare 2 different scans of the person, this is the discrepancy that we get. So the average kind of jiggling around is between 1 and 2 millimeters, which is pretty close to the resolution. So I would say that is just noise. But if we now shuffle these pairs and say, well, what happens if we register 2 anatomical scans of 2 different persons and then register the functional networks between these different persons? And then we look at the discrepancy. Then something like this emerges. We get points on the cortex where there is a lot of disconnect between anatomy and the functional role or the functional embedding of these points in the networks that are kind of related to how they communicate, how they are connected. That means a lot of the variability, we can actually kind of disentangle the reason for the variability. Is it really that the network is different? Or is it that the network is just sitting on a slightly different place on the cortex? It doesn't really matter in terms of. So Bianca Burger looked at a twin study data set where we can use the twin data in order to estimate about what part of this variability is coming from genetics, which part of this variability is heritable, which part of the variability is not coming from heritable traits, but might come from environment, from learning, from anything that happens kind of independent for 2 things. So we can now look at not only variability, but we can look at, well, is the location on the cortex, so the layout of the cortex, is that determined by genes or environment or learning? And the actual network that is sitting there, is that determined by genes or by learning? When you do that, you get a map like that. And what it very briefly summarizes is that in these red areas, we got a lot of variability, and the location on the cortex is determined a lot by genes. This variability is not coming from the experience of life that just turns everybody in a different direction. There is variability, but it is determined very strongly by genes. And so that is still a hypothesis, but our hypothesis is that that means that these areas are pretty new areas. We'll come later to how they are structured. They are variable, but they are essentially visible to adaptation during evolution because they are determined by genes, so they are still part of, so to say, an optimization algorithm because the genes are controlling them. What is not controlled by the genes is, after location is, so to say, fixed by genes, how they connect to each other. A lot of that is very similar, but a lot of variability is there coming from environment, and we could think about that like something like learning. But I think it's interesting that we see this layout is very variable in these areas, but it's also determined by genes. That's one of the observations that we can make. We'll come back to. Other ability that we gain is that now we can also look at different species. That's interesting for many different reasons, because they're model species, because we would like to understand how the brain evolves. But these different species, let's say, for example, in this case, macaque monkeys and humans, go back and forth between those two, they share some anatomy, but it turns out they also share a lot of the functional connectivity patterns. And if we use both anatomical shape and the shape of this functional connectivity, we can do a much better job at finding corresponding areas and understanding, for example, how, even though the shape of the occipital part, so the back part, kind of changes in a very simple way, what actually happens is that the vision center kind of moves around pretty independently from the anatomy because it just needs more speed. And in this case, that allowed to get a much better understanding of how these processing kind of pipelines or these processing gradients along the cortex. but I don't want to go into too much detail. But I just want to show you a couple of examples that it makes sense and there is value in kind of looking at different correspondences. Saying, well, we can follow one or the other, and we can look at the discrepancy. Yes. Yeah. Yeah. So when you look at it a bit, you see that a couple, like for example, this ridge of the motor cortex that's present in both species, and a couple of very coarse ridges are both species. But for both of these, we had functional data of many individuals. So we can look at these embedding maps and use them to align those two. So even though there is not a lot of kind of salient anchor points or anchor information on the geometry of the cortex, there is some additional information in how they are functionally connected, and you can basically use it as a feature vector to minimize the difference between the feature vectors on the cortex illustration. Very often we use these spaces to just create feature vectors that are associated with points on the cortex. Right. So a little bit time left. So I'd like to now show you two examples that are going a bit more into our attempts of understanding where is this coming from, where is this ability to reorganize coming from, where is this variability and this discrepancy. And one of the sources is early brain development. So we are very lucky that at Medical University, Daniela Breyer and Gregor Kasprian, they run a very strong group and strong clinical center for fetal imaging, so for magnetic resonance imaging before birth. That allows us to look at these babies. So you see here twins in a very early, so 18th gestational week, one baby in a much later, 28th gestational week. And the reason why they come is that something is out of the ordinary in the ultrasound, so they want to know a bit more about what's happening. They go to the MRI. But very often in the MRI, it's confirmed that everything is fine, and then if the mother agrees, then we can use this data for research, and we can use it in order to essentially try to model how the brain develops during this period. I'll not go into too much detail about it, but I'd like to show you the result, and I'm happy to discuss and provide any of the underlying methods. But essentially, we can segment the brain in all of these individuals. We can use the features of geometry, and I'll come back to that later, to match similar points on the cortical shapes of these babies. And when we talked before about the variability between individuals, it's very small compared to the variability that we observed during these 20 weeks of development from 18th gestational week to term to 38. What I try to visualize here, and that's work by Ernst Schwarz. So this is the surface of the brain in the 21st gestational week on the average across 200 individuals. And the the yellow lines are kind of the folds, the brain, how they will look like at the end of this gestational period. Just to give you something to look at, to kind of track how this changes. This is essentially the average development of this brain development over many individual brains that we observed at different points. See how the size change is totally removed here, so we normalize it. Really look at the shape. Right, so we can look at this development in the anatomical space, but we can again go back and look at functional data. Look for data that we acquire over a period of about 5 minutes. Now we're trying to more. That's work by Asena Taimotash together with Gregor Kasprian and his team. And you see, so there's a lot of movement going on, right? So there's the physiological movement of the mother, there is the movement of the baby, head movements, physiological fluctuations. So that was one instance where we thought that the PhD would be a little bit motion correction and a lot of connectivity analysis, but it actually turned out to be a lot of motion correction and a little bit of connectivity later on, but Athena managed to stabilize that and to basically create algorithms to first of all track the brain and stabilize the brain, but at the same time to also take care of the signal fluctuations that you see in the magnetic resonance imaging data when the head moves in the magnetic field. Because when you're moving in the magnetic field, you're actually generating signal, and you also have to take care of it. So if you do that, and so that's some intermediate results, so you see that's the original data, that's the stabilized data, but once you have it stabilized and once you get kind of compensate for the motion-induced signals that you are seeing over time, you are getting close to the signals that are really coming from the neural activity that is changing the dilation of vessels, that is changing the oxygenation, and that is creating signal correlations. And what you see on the right-hand side are the curves, basically mean on the x-axis you see gestational age, and on the y-axis you see how different regions start to be connected to each other in terms of we start to see correlations of these signals. One point we were really happy about was that there was a 0 at the beginning, so that meant that our motion correction worked well because we really saw no correlation at all at the beginning. Then at some point, and it's slightly different for different brain regions, we see that these correlations start to emerge. And these correlations start to emerge about at the time when we know from literature the axons start to enter in the subplate and start to enter to connect the thalamus to the cortex, the cortical areas. Before that, we knew that from histological data, we knew that from literature that looked at histological slices and actually looked at the nerves, the axons entering these different structures, but we basically were able to see that during the actual gestational aging, during the actual gestational development, we also see something that is correlated with that in terms of function. We see that function also emerges at this point and starts to show signs that might be associated with interactions. And definitely there are interactions because a lot of the connections between the thalamus and the cortex are actually responsible for designing, so to say, the roles of different areas. So now we look at these curves. I think it's again to look at the discrepancies between different areas. That's work by Anders Jakob, and he looked at a couple of networks, so 4 networks. Occipital cortex, right, so that's vision center, temporal cortex, frontal cortex, and parietal cortex. What we see is that when we're looking at when this curve really has the steepest ascent in the connectivity strength gain, we see a very stable kind of sequence of occipital, temporal, frontal, and parietal regions. This sequence of development mirrors the map that we saw before, where we had the blue areas that were evolutionary, a bit older, that were a bit more stable, that are developing earlier during the station here in this data, towards the areas that are much more variable, that are, let's say, a bit more modern in evolution, and that are also developing later here during the station. It's interesting to look. So in the closing couple of minutes, I'd like to show you the last part, which connects these 2 observations, and that is looking at evolution. And the data that we, and that's work by Ernst Schwarz. The data that we had was scans from 90 living species. And these scans were in the form of magnetic resonance imaging scans or microscopic data. So any scan where we could reconstruct the cortical shape of the cortical. There is also genetics data, and you can, and that's not really our contribution, but we just used methods that existed and data that existed to reconstruct kind of the phylogenetic tree of these different species. So you can use genetics data in order to figure out at which point 2 species were actually likely one species, and how this branching happened, and what were the approximate time points during evolution during the last time period that we are observing when 2 species split up. And the estimate, and that's why the 77 million years, the estimate is that of these 90 species, the last common ancestor was about 77 million years. Of course, there are a lot of assumptions that are just. So in evolution, and we were new to this field, the thought is that we see traits on the species that we can observe, and then we try to go back in time and see what the common ancestors looked like in terms of these traits. We can look at, for example, traits. We say, well, this species is living in groups, this species is living alone, or we can say, well, this species is kind of living on the floor, or it's living in trees, or it's living kind of below the floor, where we assume that's something that's correlated with the brain. But overall, these are all traits that we can observe in the living species, and we try to reconstruct them in these common ancestors that were there before. And the basic idea is that it's very simple, and that is that if we have a common ancestor, and at some point this splits into two different species, it's just some random process. So the thing that we are observing at the current time point in these two different species is something that's determined by some, let's say, multivariate Gaussian distribution around the point of the common ancestor. Multivariate Gaussian distribution is basically controlled by the mean of the common ancestor, by the covariance matrix that tells us something about the variability of the traits, but also tells us something about the covariance between traits. So in our work, let's say in this very simple uni-dimensional data, we can then reconstruct somehow the curves of species living on trees, or these species living on the ground, species being bigger groups, and species being living alone, because we basically go back from the observed traits to the common ancestors and can kind of iterate. And for the brain surface, we can do something very similar, but this time we don't observe this one number, like big group size, small group size. But for all of the living species, we observe the geometry of the cortex. And this data we had that for the 90 living species. We also had some endocasts, which are essentially taken from fossil records, where you see a little bit about the brain geometry. But we didn't use them for reconstruction, but just for sanity checking whether our kind of estimates of common ancestors was about right. But I think the main important thing is that we have this geometry of the living species, and we would like to go and reconstruct this path from the last common ancestor of all of these living species to, let's say, the current shape of the human brain. And the data looks like this. So these are two scans and the reconstructed cortical surfaces. And what we are doing now, and what I'll try to explain in the remaining minutes, is how we can take two of these observations and then basically work our way backwards from the observed. Just a couple of other examples to give you some sense about how different they are. So it's the human brain, that's a mouse brain, that's the olfactory bulb. We see that during evolution, how that will change very dramatically in shape and size. These are essentially all of the data examples. How much time do I have left? 10 minutes, okay. Great. Right, so this is the data. We'll talk about the colors in a second, but let's talk about the two problems that we have to solve in order to kind of work our way back from pairs of living species to common ancestors, and then from common ancestors to further. First thing that we have to solve is again the question of correspondence. What's the same point in the two different cortices so that we can work our way back to the common ancestor? We used, that's again really the accomplishments of Ernst, who worked through that. We don't have functional data for this species. That's really based on the anatomical shape of this. We have a surface mesh. We use the Laplacian and the spectral analysis of the Laplacian to create feature vector that tells us something about the local and more global geometry of this cortex. We use that essentially just as a feature point that is assigned to every point on the cortex. But then we have some additional data, and it turned out it's really important to use that because we know that it's preserved across species. So one thing that is pretty stable is our very deep sulci, so these very deep kind of valleys in the cortex that they are kind of stable across species to some extent. The thalamus area, corpus callosum area. So we used some of these points or areas on the cortex that we could annotate to give some anchors for the correspondence, and then we basically just calculated distance maps to these areas and used these distance maps as additional features that we could assign to every cortical point. We have a feature vector for every cortical point. That means that we can now use these feature embeddings, and that's a bit of a simplified visualization, I think. But anyway, we have two feature embeddings, and we can match these point clouds because they are similar enough to estimate correspondences between these two. The way we did that is something that's called coherent point drift, where we essentially use one point cloud to generate kind of a density field by a lot of Gaussians that we center around simple, and then we try to move the other point cloud in a way so that it's ending up being positioned at very high value regions in this field that is generated by the other. So we don't need correspondences to start with, but we are just moving this point cloud in this density field that is created by one point cloud, and then we take the nearest neighbor as correspondence. So we have correspondences. Now, how do we use these correspondences in order to find the common ancestor? We have these two observations, and we would like to go back to this common ancestor. One thing we have to do is to figure out, is this ancestor closer to one or to the other species? So the first step back to living species, the first ancestor is the same distance from the two living species. But once we are getting further back in the tree, sometimes one species is a bit like one common ancestor, a bit closer to the common ancestor than the other. So we are ending up basically on some point scale. And this gives us basically where between these two shapes we should position the average. But now we would like to know what's the space to think in, which space should we use in order to average these two shapes. Because if we just average the coordinates of the surface points between these two shapes, it doesn't work. It's very useless. So we operate on a space of smooth shells, and algorithmically to get the average. And algorithmically, that's actually not so complex, because what we say is instead of saying we take the point positions of the edge of the points of this surface triangulation and build the average, we take other measures that we can make on the surface of this point. The other measures that we take on the surface of these points are, for example, the area of the triangles, angles at edges, and the length of the edges. And when we form the average between two shapes of the descendants, and try to build the average to get the shape of the common ancestor, we are essentially solving an energy minimization problem where we try to find a deformation between these two shapes so that we are coming as close as possible to a shape where all the angles are the average of the angles of the descendants, all the areas are the average of the descendants, and all the length of the connections are the average. There's a optimization problem to come as close as possible overall for the shape, and take this as a mechanism to build the average while staying on this space of hulls that we would like to. Okay, so we can figure out the correspondences. The correspondences are based on the features that are coming from the surface. We can use these correspondences to create this energy function that tells us how to build the average shape. So now we have a real shape that is kind of, in our view, the average between the two descendant shapes. We can use that to transport labels or to summarize labels across two shapes, right? So if you have labels on the two descendant shapes, we can transport them to the common ancestor shape. It also means now that when we go all the way back to the last common ancestor of the 90 species, we can kind of go back through this path of ancestors that are starting with the last common ancestors about 77 million years ago to the current shape, in this case, of the human brain. And when you're looking at this tree, when you look at this tree, you see that there is kind of a little bit of difference in how much information we have. Right? There is a big travel time between, let's say, 70 million years to 40 million years where there is no bifurcation. It's definitely a limitation of this model, but then we see, as we're coming closer to the humans, we see a lot of bifurcations, and every bifurcation means that we are actually getting data and getting information how to create this shape. So this is basically traveling back forward. What you saw, this is the olfactory bulb that is now slowly vanishing, right? It's really big at the beginning, because in mice it's also really big. Vanishing, and then you see something about 10 million years ago or later at the back of the brain, and then we'll see something on the front of the brain. Now it's a very slow transition, essentially kind of between rodents and primates. Now we'll see something very quick happening at the back of the brain. We are also now seeing something on the front of the brain. Let me just any idea what this is? This is starting to walk on 2 feet. Be upright and the brain. I come to the colors. The blue color is what our estimate of where the vision center was all along. So we have data from humans and from a couple of other living species, and we can basically map it back to the prior species. The red one is the motor center, and the green one is kind of auditory parts. Yes. I put a link in the slide, so it's also online, so you can download the video. This is the one thing that when you look at the front is that there is quite a lot of inflation happening here, right? So called globalization, which is that there's a lot of growth of the front. Sorry? Exactly. So we don't have a lot of time left, so I just want to go back to two insights. One thing is that you can do is basically you can perform PCA, you can look at the variability in this overall species, set of species. Now that we have the correspondences, you can look at kind of the main modes of volume increase and volume differences and see that a couple of species, so the red ones are the rodents, the blue ones are the primates, so that they are really ending up in different areas. But it also means that we can, when we know something about the behavior of these species, like small or large group size, living in the more in trees, living on the floor, and find associated kind of volume or area increase or decrease on the cortex. We say, well, those social animals, they had much bigger areas in the red areas compared. But I'd like to show you one thing which basically comes back to this question of areas of the brain that are much more variable in the current humans, areas in the brain where there is quite some discrepancy between anatomy as a basis for correspondence and function. seems that function does not care so much about where exactly it's sitting. And evolution. So one, I think, really exciting paper was from Randy Buckner and Fina Greenan, and they posed something that they called the tethering hypothesis. They had a first estimate of this very strong globalization of the frontal cortex very recently. They just compared macaque monkeys and humans, but it's totally consistent with what I showed you. Ours is just based on more species, but you see this very rapid increase of the frontal cortex area. And when you look at these two other data sets, so this is again the Sophia Müller and Hesheng Liu data where they looked at variability. And this is data by Jorge Sepulcre, and they looked at the length of connections in different brain areas. So when you're looking at the length of connections in terms of correlation, so where are other areas that are strongly correlated with myself? And in the blue areas, these other areas are pretty close. These blue areas are clusters of points where very close areas seem to be correlated. But the red areas, those are areas where the areas that are correlated are comparatively much more farther apart. So these start to connect to other areas. This is the data. Their hypothesis was, or is, that in these blue areas we have something they call canonical networks, so kind of the hardwired circuits that do something very efficiently. And in these red areas, we have something that are much more, let's say, chaotic or areas that are connected to far apart areas. Their hypothesis is that the reason for that is that this very strong increase in the size of the cortex, it was done because computing power was needed, also led to areas of the cortex that were farther apart from the thalamus, that were less controlled by molecular gradients and also by signals from vision and auditory centers that typically structure these parts. So in animals, there is a lot of control of these parts from the sensory areas that are kind of shaping what they do. But in humans, or during this kind of period of globalization, because of the volume increase, the signals from thalamus got weaker or got less chance to reach the cortex. These areas got also farther away from the sensory areas. You could think of it as something like free computing power, very roughly speaking. So these areas started then to connect to other areas. I think that the important part of the hypothesis is that that happened, and that might be a reason that these areas now took over roles that are very unique to humans, that are integrating data and information areas that are responsible for executive decisions for things that are unique to humans. And in our data, we made an observation that is consistent with that. That comes back to the colors. So for a couple of the species, we knew what was happening in different brain areas because there was the data of marking language areas and vision areas and motor areas. Now we can transport it back to the last common ancestor, and it gets, of course, a little bit fuzzy. But we get an idea where the vision center is the blue one, motor center red, and auditory green. Now we can look at the segregation of these areas. So basically, which part of the brain is actually very close to these very specific areas, which part of the brain seems to be in between and not so much controlled by these, let's say, unimodal roles. So when we look at this measure that basically tells us how much it matters if I move left or right, how much does the functional assignment of this area changes, then if I have very crisp, clear assignments, this number is very low. So as soon as I move a little bit, I'm in a different area. When I have a lot of in-between areas, this range parameter shrinks. So it matters less about where I am because it turns out between zones. So what we would think, if the tethering hypothesis is correct, is that over time in these areas, this range grows smaller. And I'm just to make sure, I think I mixed that. So If the range is large, it means that we are in these very well-assigned areas and we are staying in the same area. If the range is small, then it means that we are getting into different areas or we're getting into this in-between. So smaller range means more of this gray in-between zones that are far apart from the sensory gradients, less controlled by them. And in the data, when you're looking at our reconstruction, with all its limitations, but we still see very clearly this decrease in this range, which in our view is consistent with the tethering hypothesis. Are areas that are becoming less specifically assigned to one of these sensory areas and are starting to be in between areas free or available to do what is unique to humans. That is coming back to the spandrels because the evolutionary pressure went into increasing the cortex size. But what happened as a side effect was that these areas were in between, they were not determined by the gradients, and they were able to develop the functionality that unique capabilities. With that, thanks a lot for the attention.